You know, usually when we think about a cancer diagnosis, there's this underlying expectation of a foreign invader.
Right, yeah, like an outside force.
Exactly. We visualize a virus breaching the cellular walls or, a rogue alien-like cell that simply doesn't belong in the body. It's a very binary narrative, us versus them. The healthy tissue is the innocent victim, and the tumor is the villain attacking it.
And, I mean, that narrative makes sense because it's clear-cut. Human beings like distinct boundaries, especially when fighting a disease. We want to know exactly what the enemy is so we can eradicate it.
But then you step into the world of developmental biology, and specifically childhood brain tumors, and suddenly that neat little narrative is just completely shattered.
Oh, completely. We're looking at a cellular landscape that is honestly mind-bending.
Right. Like, what if the deadliest childhood brain cancer isn't a foreign invader at all? What if it's just a perfectly normal, healthy biological process that simply, forgot how to stop?
Yeah, it represents the absolute definition of a biological tragic irony. We aren't looking at an alien invasion. We're looking at a profound, catastrophic case of arrested development.
Welcome to the Deep Dive. Today, we are exploring a really fascinating 2021 research article published in BMC Biology by Luo and colleagues.
It's an incredible piece of research.
It really is. So our mission for you today, our listener, is to decode the blueprint of how the brain builds its most abundant cell type, the cerebellar granule neuron.
It's quite a mouthful, but yeah.
Definitely. And we're going to look at how a revolutionary technology called spatial transcriptomics is allowing scientists to map the exact moment normal, healthy brain development takes a dark turn into a devastating type of brain tumor called medulloblastoma.
Right.
Okay, let's unpack this. We have to start with the assembly line of the brain itself, the cerebellum.
Yeah, so the cerebellum sits right at the lower back of your brain, and it is honestly a structural marvel.
I mean, just the stats on it are wild.
They really are, because while it only takes up about 10% of the brain's total volume, it contains over half of all the neurons in your entire central nervous system.
Wait, over half in just 10% of the space?
Exactly. Just let that sink in. More than 50%. And the vast majority of these are a highly specialized type called the cerebellar granule neuron.
Okay, so they're packed in there.
They are. Now, what makes this fascinating is that most of these neurons aren't even there when you're born.
Really?
Yeah, they are generated postnatally. So in a massive, explosive burst of rapid cell division during the first 2 weeks of a mouse's life, which closely mirrors early infancy in humans.
So if the brain is churning out that massive volume of cells in such a compressed time frame, how does it accomplish that without everything just collapsing into structural chaos?
Well, it requires a ruthlessly orchestrated cellular assembly line. And to understand how that assembly line operates, the researchers in our source study utilized a technique called single-cell RNA sequencing, or cRNA-seq.
Right.
They took the cerebella of young mice, specifically at postnatal days 7 and 11, which is right at the absolute peak of this cellular building boom.
The peak of construction.
Exactly. And they used enzymes and mechanical force to essentially dissolve the tissue down into individual solitary cells.
You know, I always try to visualize single-cell RNA sequencing by comparing it to a really complex mixed fruit smoothie.
Oh, I like that analogy.
Yeah, you have this blended tissue, and cRNA-seq magically unblends it. You separate the liquid back into individual intact strawberries, blueberries, and bananas.
Right.
And beyond just separating them, you can look inside every single strawberry to read its exact genetic recipe at that specific moment in time.
The smoothie analogy works beautifully for understanding the RNA isolation. By separating those individual cells, researchers were able to read the active RNA of nearly 25,000 individual cells.
Wow, 25,000.
Yeah. And RNA is essentially the messenger molecule. It tells us exactly which genes the cell has turned on to build proteins and which ones are turned off. And this whole process is driven by transcription factors, which act like master genetic switches binding to DNA to start that RNA copying process.
Hold on, though. I have to push back here. If we just look at the RNA, which is essentially a snapshot of active genes, how do you get a timeline out of that?
That's a good question.
An active gene doesn't just print out a birth certificate for the cell, right? How do we know what stage of life these cells are actually in?
That is the core puzzle. The researchers couldn't just look at one cell. They had to look at the continuous spectrum of all 25,000 cells using an advanced algorithm.
To map it all out?
Exactly, to map out what they call a cellular trajectory. By tracking the gradual shift in which genes were turned on or off across the whole population, they discovered that these granule neurons, or GNs, move through 4 distinct developmental states.
4 states.
Think of it as a biological career path.
Okay, walk us through those 4 states. Where does the cell start?
So, state 1 consists of the dividing granule neuron progenitors, or GNPs.
Got it.
These are essentially stem cell-like infant cells. Their sole biological imperative is to rapidly multiply and expand the population.
Just divide, divide, divide.
Yes. If we look at their active RNA, they're expressing cell cycle genes, heavily relying on a transcription factor called E2F2.
D2F2.
Yeah. This gene acts like an accelerator pedal, constantly driving the cell to copy its DNA and divide.
So, massive proliferation, but I mean, they can't divide forever. What is state 2?
State 2 introduces the non-dividing GNPs. These cells have applied the brakes.
Okay, they've stopped multiplying.
Right, but they haven't yet started physically moving to their final destination. They're in a transitional pause, preparing for their specific cellular career.
Like gathering their options.
Exactly. Genetically, they are defined by the activation of known genetic switches like MATH1 and BARL1, which initiate the differentiation process.
MATH1 and BARL1.
But what's really fascinating here is that the single-cell analysis allowed researchers to identify completely novel marker genes for the specific pause phase.
Wait, really? Genes we didn't know about?
Yeah, genes like TEAD2 and Srebf1. We had no idea these specific genes were orchestrating this resting state until this data laid it out.
Okay, so we have the rapidly multiplying infant cells in state 1 and the paused transitional cells in state 2. What triggers state 3?
State 3 introduces the migrating GNs. You can think of these as the restless teenagers leaving home.
Time to move out.
Yep. They have to pack up and physically migrate deeper into the dense tissue of the brain to find their permanent neural circuit.
How to do that?
To pull this off, they turn off those early progenitor genes and activate a whole new structural toolkit, utilizing genes like DCX, SOX4, NEUROD1, and notably, VIM.
Let's focus on VIM for a second, because understanding the how here is crucial. VIM codes for vimentin, right?
That's right.
How does a gene help a cell physically move?
So vimentin acts like a dynamic internal skeleton for the cell. Brain tissue is incredibly dense. For a neuron to migrate, it can't just float along.
It's not swimming in liquid.
Exactly. It has to physically squeeze, stretch, and pull itself through a thick biological mesh. The VIM gene produces structural filaments that give the cell the necessary flexibility and mechanical traction to actually crawl inward.
That is amazing. A little internal skeleton for crawling. And finally, state 4.
State 4 brings us to the mature GNs. These are the adults who have settled down into their permanent positions in the deep layers of the cerebellum.
So they're done moving.
Yeah, they drop the migration scaffolding and express genes like GRIN2B, CTN1, and another newly discovered marker in this specific context, CAR10.
GRIN2B, CTN1, and CAR10.
They extend synapses, wire into the network, and begin doing the actual computing work of the central nervous system.
Okay, let's just do a quick mini recap here. Stepping back for a second to look at the whole assembly line, we've mapped out 4 clear biological phases.
Right.
We have state 1, the dividing progenitors focus purely on multiplication, like the infants. State 2, the non-dividing progenitors gathering their options, kind of like toddlers on pause. State 3, the migrating neurons utilizing structural proteins to physically travel, the teenagers leaving home. And finally, state 4, the mature neurons that wire in and go to work, the adults.
It is a brilliantly coordinated chronological sequence.
So for you listening, think about how this mirrors a human life or a career path. Like, at what stage in your own trajectory did you stop gathering options, that rapid dividing phase, and start migrating toward your final specialty or permanent role? Every single functional neuron in your brain had to successfully execute that exact same transition to make you who you are.
It's a striking parallel. However, as powerful as that single-cell sequencing data is, it suffers from a massive fundamental blind spot.
Oh, right. Because we unblended the smoothie.
Yes.
We cataloged every single strawberry and blueberry, and we read their active genes, but the blender destroyed the glass. We completely lost the container they were sitting in. When you chemically dissolve brain tissue into 25,000 isolated cells, you obliterate the spatial architecture. You have a perfect timeline of development, but absolutely no map of physical space.
That's exactly the problem. And in the brain, location isn't just a detail. Location is identity.
Where a cell lives determines what it does.
Right. So to solve this, The researchers deployed a second, cutting-edge technology, spatial transcriptomics, or ST.
The mechanism behind spatial transcriptomics blew my mind when reviewing the source material. Like, how do we read genes without destroying the tissue?
It feels like something out of a science fiction novel.
Seriously.
Instead of dissolving the tissue, scientists take a microscopic, intact slice of the mouse brain and freeze it completely flat onto a specialized glass slide.
Okay, a slide.
But this isn't a normal slide. It's printed with a microscopic grid of thousands of tiny spots. Each individual spot is only about 55 to 100 micrometers wide.
Which is roughly the thickness of a human hair.
Exactly.
And the genius of this design is that every single one of those microscopic spots is printed with a unique DNA barcode. It's a literal molecular zip code.
It is. When that delicate brain slice is fixed onto the slide, the researchers treat it so the cells break open right where they are resting.
Right in place.
The RNA from those cells drops straight down vertically and binds to the spot directly beneath them. And when that binding happens, the RNA gets chemically stamped with that specific spot's unique barcode.
Which means when you finally sequence that RNA, you aren't just getting the gene name. Because of the barcode stamp, you get the exact X and Y coordinates of where that specific gene was active in the intact physical brain.
Yeah, it functions as a high-fidelity cellular GPS.
Wow.
By taking the 4 cell states they found with the single-cell RNA sequencing and cross-referencing them with the spatial zip codes, the researchers watched a stunning 3D map of the cerebellum emerge.
That is so cool.
They proved that those 4 developmental timelines perfectly match up with distinct physical layers.
Okay, let's lay that map out for the listener. Where do we find the multiplying progenitors?
So the first 2 states, the dividing and non-dividing GMPs, are exclusively sequestered in the outermost layer of the cerebellum. This is known as the external granule layer, or EGL.
EGL, the outer layer.
Right. When you look at the spatial map, this surface layer lights up with the markers we mentioned earlier, like MATH1 and the proliferation marker Miscayas67.
So the EGL is the nursery.
Exactly. It's a temporary biological zone right on the surface where all the massive multiplication happens.
Then those cells stop dividing and transition into teenagers. Where do we see the migrating genes light up?
The migrating genes map entirely to the middle transit zone. This is composed of the molecular layer and the Purkinje cell layer.
ML and PCL.
Yes. This area lights up with markers like MVD. You can literally track the genetic signature of these cells as they leave the EGL nursery and physically plunge inward through this middle layer.
And their final destination.
And mature genes are found deep inside the tissue in the internal granule layer, or IGL.
The IGL.
The spatial map shows intense clustering of mature markers like CNTN1 precisely in this deep layer. If we connect this to the bigger picture, fusing single-cell sequencing with spatial zip codes completely transforms our understanding.
It's not just a list anymore.
No, we don't just have a static list of cell types. We have a dynamic spatial movie of cellular migration, born on the outside, traveling through the middle, and maturing on the inside.
Okay, time for our second mini recap. Taking stock of this cellular GPS, the architecture is brilliant. We've got progenitors in the outer EGL layer, migrating cells in the middle transit layers, the ML and PCL, and mature cells deep inside the IGL.
Spot on.
So a question for you listening. If you could attach a molecular GPS to any hidden biological process in your own body to watch how it physically moves and functions in real time, what would you track? Like the precise journey of an immune cell hunting a pathogen. Or perhaps the exact mechanism of how a muscle fiber repairs itself after a workout.
It really reframes how you look at the body, doesn't it?
Complete.
But in the context of this study, building that flawless, high-resolution map of normal development was just the preamble. It was the crucial baseline needed to unlock a very dark mystery.
Right, and here's where it gets really interesting. Because you can't spot an anomaly until you know exactly what normal looks like. And the disaster they were looking for is medulloblastoma.
Yes. Medulloblastoma is an aggressive, devastating pediatric brain tumor that originates in the cerebellum.
It's a childhood cancer.
Right. The researchers focused their attention on a specific genetic subtype known as SHH medulloblastoma. To study its exact origins, they utilized a specialized line of mice called patched plus mutants.
Patched mutant mice.
Yeah, because of a specific genetic deletion, these mice are highly predisposed to spontaneously develop this exact cancer.
So they waited for the tumors to form in the mutant mice, and then they ran that tumor tissue through the exact same single-cell RNA sequencing process.
They did.
They unblended the tumor to read the genetic recipe of the cancer cells. What did the data show?
The data completely upended the traditional foreign invader hypothesis we talked about at the start.
How so?
When they mapped out the single-cell profiles of the tumor, the cancer cells were practically indistinguishable from normal, healthy, developing granule neurons. The tumor exhibited the exact same 4 states.
Wait, all 4?
It contained dividing cells, non-dividing cells, migrating-like cells, and mature-like cells.
Wait, hold on. If the tumor is genetically expressing the exact same cellular states as a perfectly healthy, developing brain, why is it a deadly mass? Why doesn't it just function like normal neural tissue?
It comes down to transition rates and proportions. This is the phenomena of arrested development.
Okay, break that down.
In healthy biology, a cell divides, pauses, migrates, and matures. It is a strict one-way street. But in medulloblastoma, the tumor cells that mimic the dividing GNPs, those infant cells in the nursery, they develop a hyperactive self-renewal loop.
So they refuse to mature.
They stubbornly refuse.
They get stuck in the proliferation phase.
Correct. Instead of shutting down the division genes and turning on the migration genes, they just keep making endless copies of themselves as dividing progenitors. The master differentiation switch is fundamentally broken.
And the source material points out that this broken switch is intrinsically tied to the SHH, or sonic hedgehog signaling pathway, which I have to admit is an incredibly wild name for a gene pathway.
The name is definitely quirky. It's derived from a fruit fly mutation, actually, but its function is deadly serious.
Right, of course.
The SHH pathway is the chemical signal that normally bathes those infant cells in the outer nursery, telling them to multiply.
Giving them the green light.
Exactly. Now, in normal development, that signal fades out after a couple of weeks, which allows the cells to transition, migrate, and mature. But in these specific tumor cells, a mutation jams the SHH receiver permanently into the on position.
Wow. So they are constantly receiving the signal to divide even when the environment is telling them to stop. The tumor isn't an alien mass. It's a mass of your own standard developmental cells trapped in an endless feedback loop of creating more infant cells. And they just accumulate into a massive tissue structure that compresses and destroys the vital brain architecture around it.
Yeah, if we connect this to the bigger picture, the cancer isn't doing anything inherently unnatural. It is utilizing a completely healthy developmental process, but it has entirely lost its temporal context.
Doing the right thing at the catastrophically wrong time.
Exactly. And it has lost its biological brakes.
Okay, mini recap number 3. Let's summarize this dark mirror of development. Medulloblastoma cells perfectly mimic normal brain progenitors, but because their SHH signaling pathway is jammed on, they get trapped in an endless loop of multiplication instead of maturing into functional neurons.
They are essentially frozen in childhood.
Frozen in childhood. So think about this for a moment, listener. What stands out to you about the concept that a deadly cancer is essentially a healthy biological process that simply lost its off switch? Does understanding the mechanism change how you view the nature of the disease?
Understanding that mechanism certainly changes how oncologists view the disease, which brings us to the ultimate pragmatic question of this deep dive.
Right. How does mapping out mouse brains translate to helping a human child lying in a hospital bed?
Exactly. We have a very elegant biological theory, but how does this insight impact actual prognosis and the future of medical treatment?
So what did the researchers do next?
To bridge that gap, they took the highly specific genetic signatures of those dividing GNP-like tumor cells, the ones trapped in the endless renewal loop.
The bad actors.
Yep. And they cross-referenced them with a massive clinical data set of 405 actual human SHH medulloblastoma tumor samples.
405 human samples. And what did the human data reveal?
The correlation was incredibly stark. Human patients whose tumors displayed a higher proportion of those specific, arrested, dividing GNP-like genetic signals faced a significantly poorer prognosis and much lower overall survival rates.
Oh, wow.
It definitively proved that these specific Peter Pan-like trapped cells are the true bad actors driving the lethality of the disease in humans.
Okay, so we've identified the bad actors driving the worst outcomes, but I have to push back here. How do you safely stop them?
That is the big challenge.
Because if these aggressive cancer cells look so remarkably similar to normal, healthy brain cells on a genetic level, how do you target the tumor without destroying the developing brain of a child? Traditional chemotherapy doesn't differentiate, right? It targets anything that divides rapidly.
That's the problem with traditional chemo, yes.
It's like trying to stop a speeding car by ripping up the entire highway system. You stop the car, but you destroy the vital infrastructure the body needs.
That is the tragic limitation of traditional broad-spectrum therapies, especially in pediatric neurology. But this is where the incredible depth of the single-cell analysis provides a totally new therapeutic roadmap.
Okay, how?
Because while the tumor cells closely mimic normal infant cells, the researchers discovered that to sustain that endless loop of rapid division, the tumor cells have to hijack and overactivate secondary cellular pathways.
Oh, they have unique metabolic vulnerabilities.
Precisely. They aren't perfectly normal. The researchers identified several pathways that were cranked into overdrive in the tumor cells compared to normal progenitors.
What kind of pathways?
They saw massive spikes in metabolic pathways, hypoxia responses, which helps the tumor survive in low-oxygen environments when it outgrows its blood supply.
Right.
And specific genetic networks like the STAT5 pathway, the ALKBH8 pathway, and crucially, the tRNA aminoacylation pathway.
Okay, let's explain that last one because it sounds incredibly dense. tRNA aminoacylation. What does that pathway actually do for the cancer cell?
Think of it as the cell's internal supply chain. Transfer RNA, or tRNA, has the job of fetching raw amino acids and delivering them to the cellular factories that build proteins.
Fetching the building blocks.
Right. Because the tumor cell is dividing endlessly, it is burning through building materials at an astronomical rate. It cranks the tRNA aminoacylation pathway to maximum capacity to ensure it never runs out of the molecular bricks it needs to keep duplicating itself.
So on the surface, the cell looks like a perfectly normal baby neuron, but its internal engine is running on a highly specific, totally hacked biological supply chain just to survive its own endless division.
Exactly. And those hacked supply chains and secondary pathways become the new therapeutic targets.
This represents such a massive leap forward. If we can design precision drugs that specifically inhibit that hyperactive STAT5 pathway or block that hijack tRNA supply chain, we aren't just blindly ripping up the highway with toxic chemicals.
The entire paradigm of the treatment shifts. The primary goal is no longer just brute force eradication of the cell.
Wait, if we use targeted drugs to block the specific pathways, keeping them trapped in that dividing phase, what actually happens to the tumor cell?
If you successfully remove the biological barrier to differentiation, the scientific theory is that you force the tumor cells to do what they were originally supposed to do. You force them to grow up.
That is wild.
By blocking the aberrant signal, you push them out of the endless loop. The cell recognizes the change, shuts down the division genes, activates the structural migration genes like vimentin, and eventually matures into a normal, non-dividing adult neuron.
We are talking about rehabilitating a cancer cell instead of executing it.
That's the hope.
Okay, let's wrap this up and consolidate what we've mapped out today. We started by taking the cerebellum's assembly line and chemically dissolving it to read the RNA of single cells, discovering the 4 strict phases of normal neural development.
We then utilized the molecular zip codes of spatial transcriptomics to map those exact cells back onto the tissue, revealing the precise physical migration from the outer nursery of the cerebellum to the deep interior as the cells mature.
And finally, we used that flawless baseline map to realize that medulloblastoma is just a dark mirror of our own development. It's a population of healthy cells trapped in the nursery, endlessly multiplying because the developmental switch got permanently jammed.
Right.
But by identifying the hijacked supply chains keeping them trapped, researchers have found the keys to potentially unlocking them. So, what does this all mean for you listening? It means that the frontier of modern medicine is fundamentally evolving. We are shifting from a philosophy of brute force to one of microscopic persuasion.
Beautifully put.
It's about deeply understanding the intricate maps of normal human biology so we can gently guide lost cells back onto their proper developmental path.
And this raises a vital concluding thought to ponder regarding how we view disease itself.
Let's hear it.
We opened the show by discussing the binary, comforting narrative of the foreign invader. But knowing what you now know about the mirrored relationship between healthy development and cancer, what if the ultimate cure for cancer in the future involves no toxic eradication at all? What if, one day, we cure a child's brain tumor simply by delivering a chemical whisper that tells the lost cells it is finally time to grow up?
It changes how you view everything going on inside you. Thank you for joining us on this deep dive. Keep exploring, keep questioning the world around you, and we'll catch you next time as we unpack another incredible mystery.